Hi friends, this is Professor R.M. Mahendrakar from JSS Arts, Science and Commerce College, Gokak. As you all know, DNA is the genetic material in all the living organisms except RNA viruses. This important biomolecule contains all the information about the organism to keep it alive and propagate. DNA carries out two important functions: autocatalytic function and heterocatalytic function. When DNA directs the synthesis of its own copy, that is, it synthesizes a carbon copy of itself, then this function we call it as autocatalytic function. If the DNA directs the synthesis of other biomolecules other than itself, like the synthesis of RNA, protein, then this function we call heterocatalytic function. In this video, I'll be discussing about the autocatalytic function of DNA, that is DNA replication. For convenience, I have divided this into two parts. In part 1, I'll explain why DNA replication is semi-conservative, and in part 2, I will explain the whole mechanism of DNA replication both in prokaryotes and eukaryotes. So DNA replication is a process by which a parent DNA synthesizes its copy based on complementary pairing. This DNA replication usually occurs during the S phase of interphase, that is just before the cell division. Now during replication, the complementary nucleotides are added on the newly formed strands. And this is just information regarding how fast the DNA replication occurs. In human beings, 3000 nucleotides per minute are added during replication of DNA. While in E. coli bacteria, 30,000 nucleotides are added per minute during replication. Now, why it is so? Just think over the structure of DNA both in prokaryotes and eukaryotes. You'll get the answer as to why in eukaryotes the replication rate is slow and the replication is fast in prokaryotes. Then the replication is very accurate. That is, it shows on an average one error per 1 millionth base pairs added. And this error also is then later rectified during the DNA repair mechanism. Replication was to figure out whether the mechanism of replication was conservative, semi-conservative, or dispersive. Now, what are these three types of replication and what happens during this type of replication? Let us see. In conservative type of replication, here when the parent DNA replicates, both the strand of the parent come in one daughter DNA, and in the other daughter DNA, both the strands are newly formed. So such type of replication we call it as conservative type of replication. Now in semi-conservative type of replication, the parental strands separate out and a new strand of DNA is secreted around each of these separated DNA strands. Thus, in the two daughter DNAs that are formed, one strand will be parental and the other strand will be newly formed. So such type of replication we call it as semi-conservative type of replication. Now in dispersive type of replication, here the newly formed strands and the parental strands are mixed randomly in both the daughter DNA molecules formed. Now a question arises as to out of these three, which is the type of replication that occurs in all the living organisms. Now, this was solved by Matthew Michelson and Franklin Stahl, who set up an experiment in the year 1958 to prove that uh, correctly which type of replication occurs. Now, their idea was to make the parental DNA molecule of heavy density by culturing them in a medium containing heavier isotope of nitrogen, that is N15, and make this heavier DNA containing cells to replicate in a medium containing nucleotides of low density. So their prediction was, now suppose if the DNA replication is conservative, then when this heavier DNA molecule, when it was grown in a medium containing the normal nitrogen, that is N14, now here the parental strands will be both together in one DNA and the other, both will be newly formed. So the newly formed DNA molecules would be having N14 nitrogen, and hence its density would be lighter. So thus they would be getting two bands, one heavier band and one a lighter band. If the replication was semi-conservative, then in the first generation, when the parental DNA separates and it synthesizes a new DNA molecule, now here the parental strands will separate and the new DNA strands will be found on each parental strands. So both the parental strands, both the daughter strands would be having one parent DNA strand and one newly formed DNA strand. So that means one strand would be having N15 nitrogen in it, and the other strand would be having N14 nitrogen in it, and hence the density would be intermediate, and thus they would get an intermediate level in the first generation, and in the successive generation they would get intermediate and lighter if it was semi-conservative. Now, if the replication is dispersive, now in dispersive mechanism, we have seen that the newly formed DNAs and the parental DNAs would be mixed randomly in both the strands. So in this way, what would happen? The N14 and N15 would have mixed in both the daughter DNAs here, as you can see here, and thus the density would be intermediate, and in the successive generation also it would be intermediate. So these were the three predictions. Now, based on these three predictions, they performed this experiment and finalized that which type of replication occurs in all the cells. So with these predictions in mind, they carried out their experiment in the following manner. They allowed E. coli cells to grow in a medium containing the heavier isotope of nitrogen, that is N15, for 14 generation. This isotope was inserted into the nitrogen bases, which then were incorporated into the newly synthesized DNA strands. After 14 generations in N15, the DNA of the cells were labeled with heavier isotope. That is, both the parental strands were with N15 isotope of nitrogen, and this they confirmed with this cesium chloride density gradient centrifugation. So when the centrifugation was done, they saw that the DNA accumulated at the bottom level, indicating that the density of the DNA was heavy and both the strands were containing the heavier isotope of nitrogen. The cells were then removed from the N15 medium and put into a 14N medium. After 1 and 2 cell divisions, samples were taken and the DNA was isolated from each samples, and the DNA was examined for the radioactivity isotope of nitrogen through density gradient centrifugation using cesium chloride. Michelson and Stahl were able to distinguish the DNA of different densities because the molecule had to be separated from each other by a procedure called cesium chloride gradient centrifugation. If cesium chloride is spun in centrifuge at a tremendously high speed, that is around 50,000 revolutions per minute for many hours, the cesium and chloride ions tend to be pushed by centrifugal force towards the bottom of the tube. Ultimately, a gradient of ion is established in the tube with heavier DNA at the bottom, lighter DNA at the top, and hybrid in between. So after each generation, the DNA cultured in the N14 medium was isolated and centrifuged by the cesium chloride density gradient centrifugation technique, and this result was obtained. That is, after first generation, they found that all the DNA accumulated in the intermediate region, that is the hybrid level, indicating that one strand was of N15 and the other strand was of N14. Then in the second generation, when the DNA was isolated and centrifuged, they saw that 50% of the DNA was in the intermediate region and 50% of the DNA was in the lighter region. And in the third generation also, when the DNA was extracted and analyzed, they saw that 25% of the DNA was at the intermediate level and 75% of the DNA was at the lighter level. Now, these results that were obtained by them clearly indicates that only semi-conservative type of replication occurs in these cells. Now here you can see when in the first generation, when the DNA replicated, now the parental strand was with N15 and the newly formed strand were N14. So both the strands were having N14 and N15, and hence they were intermediate in density, and hence all the DNA had accumulated in the intermediate region. Now in the second generation, now when these DNA separated, now here the strand separated, so one strand was having the parental N15 and N14, and here the both the strands were with N14. So similarly here, so thus in the second generation we got 50% with intermediate density and 50% with the lighter density. And in the third generation, when this DNA replicated, okay, 25% of the DNA was intermediate and 75% of the DNA was lighter. So these results clearly indicate that the replication of DNA is semi-conservative.